All right, so we just have a few more slides that we're going to quickly go over. Most of this part here at the end of chapter 11 really should be a review of general biology. And so we are going to go fairly quickly. What happened? Okay. It's not letting me make that one any larger. So these are the different ways of transporting. So now we're talking about membrane transport of items. And so in the beginning, or up here at the top, let's see. I was really trying to see if I could get that to. We have facilitated diffusion. Okay. And so facilitated diffusion, does it require energy? Ignore that. So does it require additional energy in order to remove the solute from the outside to the inside? No. Okay. Then we also have, we'll talk about active diffusion. We'll go into greater details in just a moment, where that one does require some type of energy input. Many times we're talking about the hydrolysis of ATP or GTP. We can also have secondary, which is where it's coupled active transport, where things can either be coming in at the same time for symport, or they can go opposite directions for antiport. Then we have our ion channels. You can have an ionophore, which may be that's the one that usually people forget whenever they're just listing it. And then finally, if it's a small molecule, then it's a simple small hydrophobic molecule. It's a simple diffusion where it can go according to the gradient from outside to inside, or vice versa. All right. And so for the transporters, I like this flow chart that's in your book just because of the fact that it divides it up. It's pretty simple to follow. You can imagine that there are two broad classes, the carriers versus the channels. Okay? And so a carrier is one that's going to be aiding the solute to get across. And so you can either have it be active, a primary active transporter, or you can have it be this passive or secondary active transporter. Okay, and so we're going to go over and see some examples. In particular, we're going to focus primarily on the sodium-potassium channel, because it's the one that's most likely the best well characterized. So, as I mentioned before, passive transport doesn't require any additional outside energy. It's driven solely by the concentration gradient from either inside to outside or vice versa. There are two types. There is a simple diffusion, which is where it moves through a channel that's created by some type of pore and protein. Or you can have a facilitated diffusion, which is carried across by a carrier protein. And we'll see that some next semester when we talk about some of the carrier proteins and the shuttles, in particular with respect to the mitochondria and the way that things get differentiated between the different portions of the mitochondria. And so, well, come on now. One way that you can characterize this kinetically is that if you just look at the rate of transport as a concentration of whatever it is that you're trying to transport, the solute, if it's simple diffusion, it's linear. And so it just keeps increasing as you go add more solute to the exterior. Whereas if it's facilitated diffusion, you're going to see a hyperbolic function. Okay, and why is that? Why will it level off if it's facilitated diffusion?
There's only so much you can facilitate.
Right, there's only so many enzymes or pores or whatever it is that's doing the facilitating. And so therefore, at some point in time, at some concentration of solute, it's going to be completely bound or busy, when you think of it that way. And so therefore, it will level off. And I'm not sure what happened to the picture there, but wait a minute, let me see if I can. Whoa, there we go. And then you can see with passive transport, we have the facilitated diffusion is one where you've got to have some type of conformational change or aid in the sense of getting the solute across, whereas if it was just simple diffusion, you can either have a small molecule such as oxygen just going across the membrane on its own, or you could have something, that can flow through the pore itself, which I'm trying to think of a good example of that off the top of my head. No. I was going to say maybe perhaps some types of, like water molecules and things like that. Okay, whereas active transport, it's moved against the concentration gradient. And so, for example, we're going to talk about the sodium potassium transport in just a moment. And so this is just to show a little cartoon. I don't remember where I plagiarized this from, but to show how it's going against the gradient in order to get more of the minerals or whatever it is across from one side to the other side. Okay, and almost always we're talking about, in this instance, we're talking about the hydrolysis of ATP. Now, when we do the signaling cascades, it's not really membrane transport. Many times then we're talking about GTP, but here we're talking usually about ATP. And being hydrolyzed down to ADP. Those next semester we'll also talk about how there are different ways to hydrolyze the ATP itself. And so the example that's probably the most well characterized is the sodium-potassium ion pump, or the sodium-potassium ATPase. And we'll go over the different steps with respect to it. What is it used for? Where do we see the sodium-potassium pump?
For filtration.
Right, we're talking about some type of impulse. And it's because it creates an ion gradient in itself. It's not 3 to 3. Oh, I lied. There's one more thing. And I've already used these terms. There's uniport, which this is one molecule going one molecule going at a time. Or if you have multiple molecules going, usually we're talking about 2, then you can either have them going in the same direction, which would be sim port for the same, or anti-port means they're going against, they're going in opposite directions. Here we have the sodium-potassium ion channel, which to me, I don't know, it always reminds me of a nutcracker, which is, what?
A lobster claw.
Oh, a lobster claw. I could see that coming from someone from Maine. So, okay. But, so what happens first? If you notice, it's a 3 to 2 ratio. And so, whenever the transporter, if we start here, whoa, not like that though. If we start here, whenever it's open for the inside of the cell, on the cytoplasmic side, then we can have 3 sodium ions be bound. Okay, then ATP is hydrolyzed to form ADP, and when the enzyme itself becomes phosphorylated, the lobster claw, I guess, then opens up the other direction, and those 3 sodium ions can escape to the extracellular side. Okay, then the binding site for the 2 potassium, or I should say the binding sites for potassium, there are only 2 of them, so 2 potassiums then go on the inside. You get hydrolysis of the inorganic phosphate, because remember that's what that PI stands for, it's inorganic phosphate. It's not like on ATP or on sugar or anything like that. And the hydrolysis of that is what causes the lobster claw then to open up once again in order for the 2 potassiums to come out. And so if you notice, there's a net gain of a positive charge on the outside of the cell versus the inside, so it's going to polarize the cell. Now, later on, whenever you're going to have this like muscle contraction or the nerve impulse or what have you, then it becomes depolarized. And so then the other channels will open up, the sodium will rush in, potassium can rush out, and then you have to do the repolarization through the same process here in order to get the overall net positive charge on the outside versus the inside of the cell. Okay. And actually, I've already discussed this, but I wanted to review it once again, is if we think about many of these channels, in fact, all I can think of off the top of my head, are going to be polypeptides, multi-proteins coming together. Many times they're alpha helical. And so we've discussed this somewhat, like what kind of interactions can help hold these proteins together with respect to the membrane itself. So what kind of protein, what kind of interaction? So if we have, whoops, the draw. Ooh, it gets really fuzzy. Okay, so what kind of interactions can go between the two helices? Pardon? You could have hydrophobic, okay? And then especially for the exterior of it, why would that make sense? That this portion here and here would be hydrophobic. This is the way that I have it drawn. Why does it make sense that that's going to be hydrophobic for the membrane protein? Pardon? It's on the inside of the membrane, okay? What kind of interactions can happen between the two helices here to help hold them together? You can have hydrogen bonding, and you could also have, what's another possibility? Electrostatic. Those are also, the old term for it's called a salt bridge, because you have a positive and negative, which technically that's what a salt is. And so if you just think about the properties of the alpha helix, to where if I take those alpha helices and right now they're long ways, but if I look down them like you're looking down the barrel of a gun, once again, please don't look down the barrel of a gun, but it's going to look something like this. Whoops, it's going to look something like this. Actually, that's not too shabby. Okay, so faith integration too. It's going to look something like that. Okay, and this is called a helical wheel because with each turn would be a new amino acid, so to speak, on the outside or around that turn of the helix. And if you just look at that, and many times what you'll notice for these, just like we've done the hydropathy or the hydropathy index or the hydropathy plots, which we just had on that quiz, they can do these helical wheels. And you can see that they are amphipathic, and that's what the next slide shows. So here's just one alpha helix that's going to be in the membrane, and if you notice, see, it kind of looks like the little wheel that I've been talking about. But we have this side, what does asparagine, serine, and threonine have in common versus leucine, phenylalanine, valine, isoleucine? These are polar, so that the dark side is polar, the lighter side is nonpolar. And so therefore, you can imagine that if there's another helix in this multi-subunit complex, or even a really, really large protein where it's going to have multiple helices what's the word I'm looking for? Transverse the membrane, then you can imagine that this portion here, whoa, I didn't mean to do that. This portion here on the next helix would be polar as well. And so that way is what's going to hold the two helices together. And so it's really helpful in also looking at the way that the helices can come together in multi dimensions. Okay, and so that's why, just like there are bioinformatic apps and programs now that will take and will do the helical wheels for you, so that way you can see, because it knows what amino acids tend to form alpha helices versus beta sheets. And so it'll take the regions of your protein, show you what regions are alpha helical, and it will run these helical wheel diagrams and show you, like, okay, which sides, like, do you have any that look like they're more polar than versus non-polar? Just like there are apps and programs that'll do the hydropathy plots for you. Many of these are free, and so they're useful whenever you're first trying to characterize your proteins, your protein of interest. So that's the end.